Hi, everyone. Welcome back to Cells to Cell. My name is Heem. That's Nick. And our very special guest today is Dr. Evandro Fang. Dr. Fang is a associate professor at the University of Oslo in the Department of Clinical Molecular Biology. He's also the coordinator of three different things. One of them is the Norwegian Centre on Healthy Aging Network. Another is a coordinator of the Hong Kong-Nordic Research Network. And the third one is he's the coordinator of the Norwegian Alzheimer's Disease Network. So now we'll give Dr. Fang or Evandro just a minute or two to talk about his work, and after that we'll just get into every question we have about him.
Yeah, thank you guys for having me. I'm now an associate professor in the University of Oslo and also affiliated to the Akershus University Hospital, which is the largest hospital in Norway. And My background is I had a bachelor and a master degree in preventive medicine. Then I went to Hong Kong for a 3-year PhD from 2009 to 11, focusing on drug development. It was my time in Hong Kong, a small city with condensed population over 7 million. And I got the impression in 2010 that the mechanism of aging could be a very interesting topic, but not very well explored. And then I was driven by this aim and went to the National Institute on Aging in Baltimore, close to Johns Hopkins, for a 6-year training on molecular gerontology with two esteemed professors, including Wilhelm Bohr, a Danish American professor working on DNA repair and aging, as well as Professor Mark Madison, who worked at NIA, now also affiliated to Johns Hopkins University. And after that, I came to Norway around 6 years ago. So there are three topics in my group. The first one is the biology of aging, focusing on why we, reduce the muscle volume, lose the memory, and lose the hair. And the second topic is age predisposed dementia, focusing on Alzheimer's disease. And the third one is the use of artificial intelligence to prepare drug development. And those work have been performed by a total of around 15 students in the lab, coming from like 8 countries. So it's a very international team.
That's incredible. That's incredible. Three really, really focused yet broad topics that are going to be very, very useful in the next, they've already proven their use, but they're going to become more and more prevalent as regenerative medicine and stem cells just become more used in medicine. And I think by you hopping on the ball a little bit earlier is going to really make your lab stand out. We were talking early, before we started, about you winning the national prize for your Alzheimer's disease research, and how cool that was. So, your lab's doing great work, and it sounds like you're at the University of Oslo, which is just an unbelievable research facility. So, how go a little bit more in depth for us if you could. Like, how did you get interested in studying aging and how you got on this path of going to study at the NIH? And now that you've landed at the University of Oslo, how did you really get fixated on this topic?
Yeah, as I mentioned briefly in the self-introduction, I think my passion to working on the biology of aging stemmed from my PhD experience in Hong Kong. A city can host over 7 million people, famous for those skyscrapers. And actually, the health service in Hong Kong is very high. So, and the average lifespan for both men and female are very high also in Hong Kong. So it makes there are even like over 12 years ago, we already see there are so many populated elderly in Hong Kong. And many of them needed to be dependent on wheelchair, as well as a lot of do not have good memory. And which makes me to think about as a trained medical student, how I can contribute to enable them as well as us, because all of us will getting older one day, to have a healthy, happy, both physically independent and mentally sharp life. So with all those things in mind, I thought I need to change my research focus, not just on drug development, but to understand the aging process at a molecular level. Then from the end my third year of PhD, so I start to send like my job applications as postdoc to different professors. And the one which I'm interested in, and also I got the offer, was from Professor Wilhelm Bohr. So he came from Denmark. Both his father and grandfather were Nobel Prize winners in physics. So his grandfather was Niels Bohr, as famous as Albert
Whoa.
Einstein, and who was also involved in the Manhattan
Wow.
project. Yeah. So I think inspired by him, he said, Evandro, I brought you to Baltimore. I want you to think big, not focus on small things, but what things you are going to do in the coming years can potentially or very likely to be translated to the clinic to benefit to those people suffering from aging as well as pathological aging. So I have trained with him and market medicine for 6 years, but definitely we have been very productive in collaboration with another postdoc, Morten Schreiber Knudsen, now also a rising star in the aging field in Copenhagen University. We had a lot of joint publications in cell metabolism, Nature reviews, and those publications enable me to be competitive to find a job in the University of Oslo 6 years ago.
That's amazing. And if we focus a little bit more on the specifics, so one of your funding opportunities I saw in VitaDAO was more about mitophagy, right? So that's
Yeah.
about your mitochondrial dysfunction. So I guess we could talk a little bit about that and how mitochondrial dysfunction is the reason a lot of aging happens. And you have identified two main primary compounds. If is that right?
Yes.
And how did you use AI to did you find these two primary compounds with your machine learning platform first, or did you develop everything kind of together?
Yeah, it's a gradual developed idea. So my interest in Alzheimer's disease started from 2015, like 3 years after I started my postdoc when I was still in Baltimore. If you look over Alzheimer's disease patients, many of them won't get the disease until their 60s to 70s. By using the post-mortem brain samples from Alzheimer's disease, we find there are a lot of tau and A beta. We consider they are the garbage accumulated in the brain. And we hypothesize in the field, not only us, that is accumulation of those garbage is the cause of memory loss. And my group is very interested in why the garbage clearance system, like the garbage truck, does not work anymore. How we can find a way to solve the problem. So when we dig into the details, we found that the garbage truck actually worked very well when those dementia people were young. However, when they are getting older, the engine of the garbage truck worn out and does not provide energy to the truck. So the truck, even it is there, it does not work. And it leads to one of the reasons to cause the garbage accumulation. If you consider the engine of a garbage truck, which is the mitochondria in the human cells. So when we are young, normally we also have continuously damage of mitochondria, but they can be efficiently recognized and repaired by a process called mitochondrial autophagy, termed mitophagy. So the long story in short is the engine in our human cells, the mitochondria, can be continuously damaged, but they can be efficiently repaired through a process called mitophagy. However, when we're getting older, especially in Alzheimer's disease, this process does not work, and finally results in the trunk does not work and accumulation of garbage, and finally memory loss. And we used 5 years to validate this hypothesis is right in collaboration with our colleagues in Oxford as well as in NIA, among the other institutes. So once this concept is proved, then I start to think how we can find approach to repair the damaged engine. Are there any small molecules we can use to repair the engine? And in collaboration with my colleagues in University of McCall, which has a small library, contains over 3,000 compounds, isolated from traditional Chinese medicine, which has been proved to treat dementia before, but which molecule they do not know. If you have a full-time PhD or postdoc to screen 3,000 drugs, if using cell culture and C. elegans as the platform, it may take us more than 3 to 4 years to do the drug screening. So that is too long. And in these modern times, I think we need to use, the advanced technology. And while I have very basic knowledge on AI, but I have excellent collaborators in Imperial College London and a company called Rank AI. So we discussed. So here I provided them like around 19 mitophagy inducers which can repair the engine. And I asked my colleague, can you using AI to to have an algorithm on their structure or anything else to find the similarity? Then we use this algorithm to define any potential mitophagy inducers in this big library. And we only used 5 months to build the algorithm. Then another few months for the drug screening and finish the whole project in 1.5 years. So it's dramatically reduced from 4 years to 1.5 years. And we were so lucky the paper published in Nature Biomedical Engineering, and we got a lot of positive attention. So this paper enabled us to identify K and R, those two molecules. And we had foreign collaboration with VitaDAO on for the structure modification, as well as to working on new additional compounds from passion fruit in collaboration and supported by VitaDAO and Molecule.
That's amazing. And congratulations on all that progress and all your publications in such reputed journals. So I guess my question would be, are you familiar with like the molecule NMN, so nicotinamide mononucleotide? People use it as like a dietary supplement because it's a precursor to NAD plus, which is quite literally the subunit that mitochondria is used for pure energy. So have you like looked into using NMN in any of your research or anything like that?
Yes, I think you are asking the right person in addition to David Sinclair and Charles Brenner. So when I joined the Wilhelm Boss group in Baltimore 10 years ago, so in general, in this field, for the NAD precursors, there are 2 major molecules. The first one is nicotinamide riboside, which was identified by Charles Brenner in his 2004 Cell paper. And he then patented the way to produce NR and has built a company called Chromadex, which provides those NR molecules to the researchers to do preclinical and clinical studies without charge. And we are actually the first one through its like material transfer agreement when I was in Baltimore. So we did a lot of experiment using NR as approach to treat premature aging disease as well as Alzheimer's disease. And those studies enabled us to publish a paper showing that NR actually can change, goes to NAD plus, and it can be a mitophagy inducer to repair the damaged engine of the garbage truck as a way to increase the garbage clearance. And this further enabled us to do a clinical trial on ALS as well as premature aging disease. I'm not directly involved, but Wilhelm Boy is involved in an NAD-based clinical trial in Norway. And the second camp is Professor David Sinclair. So he is major leader on NMN, nicotinamide mononucleotide. NR can convert to NMN, then NMN to NAD+. So NMN also works very well in our hand, especially in cell culture, as well as when you are working in worms. NMN seems to be much more stable than NR, although both of them show similar benefits. So we have also been working with NMN in collaboration with David Sinclair, and we published one paper in cell metabolism, one paper in, two cell metabolism papers.
Wow, that's really awesome.
Yeah, that's great. That is really great. When you are, working with these small molecules and you find an analog that works, what assays are you doing to test these? You mentioned cell culture. Are you doing any in vivo? And kind of, can you just explain how you're getting, all these great positive results?
Yeah. So definitely, I think after this meeting, I will send you a recorded presentation I gave to my colleague a few period ago, so you'll know more details. Now I would like to share with you and the potential audience what we normally do. First, there is an activity assay to see whether this molecule can induce mitophagy as a potential to repair the engine. Once we found those molecules can induce mitophagy, then we started to do functional experiments. For example, we have both C. elegans and mice. When they overexpress Alzheimer's disease related risk genes, then they show memory impairment. And we treated C. elegans and mice with a drug of interest for a period of time. Then we followed by the memory experiments. I want just to share with you how we use. Have you guys heard of C. elegans or the worm before? Do you know what it is?
Mm-mm.
It's a tiny worm, 1 millimeter. It's like a comma when you write your papers. It's very small. It is transparent. And the worm actually, studies on worm enabled 3 Nobel Prizes to be awarded to scientists in the field. I think the first one, it is the sequencing of the whole genome in an animal that started in C. elegans by the grandfather of C. elegans, whose name is, I forgot the name, but he got the Nobel Prize. And the second one is the using of the GFP or FP awarded to some professors. The one used C. elegans as a model to express GFP protein in worms. The third one is RNA interference. So those are so fundamental in the biology field, were started from C. elegans. Using C. elegans, for example, how we do memory in C. elegans. Because this is a tiny worm, they can live only 2 to 3 weeks, but they have a human brain-like structure, and it will die in 2 to 3 weeks. So they are excellent model to do aging and memory research. So how we tell ourselves that this animal has a memory or not, because they cannot speak. So the concept is the worms like to eat food all the times. And when we move the food away from the environment, transfer the worm from a plate with food to a plate without food. In an environment without food, we put a type of smell, like odorant, like popcorn smell. Then the worm will correlate. Popcorn smell means no food. Then you bring the worm again to a new plate, and also with popcorn smell. So, but without food. If they can remember, then they will go away from the popcorn smell area because they correlate with no food. However, if they cannot remember, they still be attracted to the popcorn. So through this negative correlation, then we will tell ourselves after a specific mitophagy drug treatment, it can improve the memory or not.
Wow, it's a very interesting way to look at all that thing. And
it's smart design.
I was gonna say, who would ever use worms for memory testing? I think that's wild. Everybody thinks you would use a mouse in a maze or something, but yeah, I'm sure that these worms. So how hard is it?
Nick, just one point. Normally, after we finish the screen using the worms, we still wanted to use a mice, like different type of Morris water maze or Bunny's maze, to validate our results. However, in a practical life, everything costs the money, everything costs the time. And there are very strict ethical regulations on how many mice you can use. So it's nice to start with C. elegans, then to validate in mice.
So how do you initially treat these C. elegans worms? How like because with like I have the opportunity to work with mice. I also work in like a drug development lab, and we focus on like the hematopoietic system. But injecting a mouse is really easy. You just do
Yeah.
it subcutaneously or intraparenterally. But like, how do you distribute the drug to these these 1 millimeter worms?
Because the worms, they are so small, so we all normally using a transparent Petri dish with a diameter like 6 cm. Then we at the bottom of the Petri dish we put the agarose there or agar-based solid gel there. Then on the top of it we put E. coli OP50 as a food to feed the animals. Once we start to giving the drug treatment, if the drug can be dissolved in water, we can just add on the top of the agar, let it diffuse for 1 to 2 days to be equally spread. If it does not dissolve in water, then we dissolve in DMSO and add it at the time when we prepare the agar at the temperature of 55 to enable those drugs won't be degraded due to high temperature.
That's pretty cool. That's really, really cool.
I had a question more about like all your funding sources, and then actually I had a question also about like your how like kind the social scene is in Norway when it comes to longevity and anti-aging, and if there's like government incentives in place for people to live a healthier life. Like, do they give stores like, I don't know, better incentives than like here in United States? We don't have a lot of like fast food restaurants, for example, that really give us that cater towards like healthy living, and these are all just things that like are longevity. Um, but like, how is it in Norway? Do you find like a lot of the social scene is like toward more longevity oriented?
Definitely, I think if you compare Baltimore to Oslo, they're almost, except to in the hospital. When you walk on the street, I think most people are very in fit. Not, I think in the USA we have so many overweighted and big individuals.
Yeah.
This is a little bit sad, but it is the case. Uh, I think there are a couple of reasons in Norway they are in good shape. Norway is traditionally a country they highlight the importance of balance of work and private life. So in the weekend, they normally go to the cabins, go hiking, go to the gym rather than just sitting somewhere to eat the food. And secondly, I think there's a culture here, especially we have a long winter. So they encourage the traditional Norwegians to go out, to go skiing, a wonderful way to keep fit. And Norway is not belong to EU, but is an EU affiliated country. Actually, Norway has very strict rules, even higher than the European rules regarding, the hormones in the chicken, how to raise the chicken, the daily food, the quality is even better than many of the European sold products. And number four, for example, actually in Norway, we do not have KFC. We have McDonald's. Do you
Really?
know why? I give you
No.
guys a chance. Yeah.
So you said why they have McDonald's and not KFC?
Yeah.
Does it have to do with the mass production of the chicken or something like that?
Yes. I think it says the way, so the chicken KFC use does not qualify for the Norwegian standards. Yeah. Because of cage, because of the amount of, what type of food they fed the chickens. It really says something. Yeah.
Mhm. Do you do skiing? Do you go skiing and everything?
I think I like skiing very much. So I try to use this flat one because when you do the flat one, you won't fell down or especially you are not that so young. And the most time I do is the badminton. So we actually Denmark has been very famous in badminton, for a long time. Norway and Denmark, they are brothers, so there's the positive influence to each other. I play like badminton 2 times per day and do a lot of exercise and go hiking in the weekend. And if you follow our Twitter, you'll see we have a lot of active outdoor activities. Many students in the lab, they do fasting. They like to go outside for a hiking. So we arrange many of such activities.
So these students in your lab, are they like what, how old are they? Like, are they undergraduates? Are they graduate students? Are they PhD candidates? Like, or is it just a big old range of, like you said it was people from, 15 people from 8 countries.
Mm.
I'm sure the culture is awesome in the lab, but you know, what do you look for like in your employees to come in and help you with this great work? And I'm sure, have fun both in and out of the lab.
Yeah, we have 5 plus 2 PhD students. The number 6 just came from Spain. Number 7 will be in position by the end of this month from Austria. They are, at their middle to late 20s, and then have 5 postdoc or fellow. They are at their late 20s to early 30s, and a couple of master students and the lab manager. I think the key thing for them to come to Norway, many of them actually from very prestigious university like UCL, King's College London, Imperial College, and some from Denmark. I think one of the reasons for them to come to here is they're all interested in the biology of aging, as well as the mechanism of Alzheimer's disease. And this attracts them from different European countries, as well as one guy from USA joined us and just graduated, Jared Rice, if you look over my website. I think they are interested in the work we are doing. That is the key information.
I wanted to go back to like MoleculeDAO and like VitaDAO. So how did you first start your relationship with them? Did the organization form and then they found you based on your publications? Or did you see that they were doing funding and then I guess talk a little bit more about the IP NFT thing
Yeah.
and like how that all plays into your whole lab?
Yes. I would say right now it's the best time to do agent research. Speaking of the funding, in addition to the classical funding resources, there are so many private and new foundations which are giving fast and decent money for the agent researchers when you have good ideas and you have good tracking record. And compared with the classical funding agencies, those new ones, they are less demanding. They just ask you to write down like 2 pages rather than classical 10 to 20 pages. And they give you a quick feedback from application to the like result release, maybe only 2 months, compared to classic one can be half a year to even 7 or 8 months. So it's very attractive to the busy researchers. And speaking of the case of VitaDAO and molecules, they collaborate with each other. I think I saw their information in my Twitter, so that was the first thing. And then they contacted me because they are interested in drug development, and we have some decent ideas on how to repair the engine of the garbage truck, and which they really like these ideas. And they asked me to write like 2-page proposal. And then I wrote it and I had an interview to the board members. And I convinced them this is a good idea for them to fund us. But I want to highlight one thing. It is the intellectual property for VitaDAO is a little bit different from the other foundations, and VitaDAO claim all the intellectual property. So there's pros and cons of this part, but we thought this very well. This is a new concept to the Nordic countries. However, I think the good thing is, especially we want to do commercialization. For example, even University of Oslo has its own spin-off company to help the PIs within the university to do commercialization. But there are so many projects as well as they are not so, they are more familiar to European laws and rules rather than the Americans, as well as less solid networks compared to VitaDAO. So the good thing for the VitaDAO is they are so determined on commercialization. They have very solid networks to the vendors, to the potential pharmaceutical companies who may want to buy the patents. And then the university has, and VitaDAO has a good agreement. If I give you the fully intellectual property, what I should benefit if one day it can be purchased by a company to do clinical trial. So they got a decent sharing of the benefit between the two parties.
So do they take care of all of the applications for you? So for all the patent applications, so they're just like that agency that takes care of all of that. You don't have to do anything through the university.
They take care of everything. So that is the wonderful for us, because we do not know how to do it, and they know everything and they save our times. Yeah. So I'm very, very happy to collaborate with them. Like I think I'm the second one to be awarded by VitaDAO.
Wow, I think this initiative is so great because I think the future of medicine is definitely these small molecules that are highly targeted and they're very well researched, and also like cell therapy, gene therapies. I feel like this is all going to be the future of like medicine in the next decade or so. And I guess that would be my next question to you is what do you think the future of medicine is headed toward? Like, because I've worked in a lab that uses nanoparticles, so it's like highly targeted for drug delivery. So how do you think all of this will come about, and which countries do you think like are I know in Europe for food especially they don't like any GMOs, so genetically modified organisms, nothing of that sort. But we don't have those regulations here in the United States. So how do you think a lot of these therapies will come about in like the European Union and also Norway?
Yeah, this is a question a little bit out of my comfort zone, but I can share with you my limited impression. I think there are, because there's, the way to improve people's quality of life, the way to treat different diseases, because the difference of different people, the difference of genetic reasons, the difference of the like a disease, whether it's a skin disease or it's something with inside of the deep of the brain. So you need to develop different approaches, either to do a direct surgery or chemotherapy, or this is small ultrasound-based treatment or small compounds. So that is common. We develop different methods to target on some specific health issues we aim to solve. This makes this field is very interesting and highly competitive. And speaking of the rules, whether European or US, the difference in the approval to sell genetically modified products. So those are the rules based on different countries. It's hard for me to give comment. But my general understanding is we must to make sure it is safe, not only for short term, but also for a long term. A lot of new products, they have the test for a short term like effects without any side effects. But how about long term? For those things, maybe after 10 or 20 years, when we get more clinical information, then we'll have a better regulation on such products. And there may be a unified agreement between Europe, Asia, and USA on how to regulate such genetic modified products to the markets.
what are some of the most important lessons you've learned so far in your whole journey in terms of team building, in terms of what to look for when you're developing these things? And do you just have like a natural eye to look for when you say, okay, this is interesting data from the
Mm.
lab, I think we should focus on this more, versus something that you feel like is not that important and you could potentially waste funding towards?
So before I start that, I want to ask you two, how do you foresee yourself in 10 years? Where you will be in 10 years?
Oh man.
Go
Me,
ahead.
at least, yeah, I'll go first, I
Yeah.
guess. Um, I guess for me, I just wanted to, I'm like more interested in like the entrepreneurship and business part of like the whole medicine thing, and I'm very interested in like translational medicine. So in
Mm,
10 years, hopefully I have the ability to help some startups come about, get acquired, or have a company of my own where I'm doing something, definitely in the whole regenerative medicine field with cell therapies or gene therapies. So
mm,
that's like where I want to be in the future. Not direct, like you're very involved with the science, like you're the brains of this whole operation. But there's got to be a team that makes everything work, right?
yeah,
So I want to be that person that kind of makes it all work in the commercial space.
I got you.
Yeah, that's the same
Yeah.
way that I am. I'm very much so interested in the science, but I see myself going more of like the business and product development side of biotech. So, when I graduate in, May, like I hope to set up something that's in product development or business development. I had the opportunity to work for an umbilical cord blood bank as my
Mm,
internship over the summer, and I did product development with them, and it was an absolute blast. I learned so much, but I'm also the same way with Heem. I want to also be the developer of something or create a team that develops something really cool, run it, kind of be like a CEO, something,
mm.
where I kind of have power's the wrong word, but I really like having conversations that can change directions and really change people and change their lives. And, my dream is to run my own company one day. And even if I don't get acquired, I just want to run something and make something that's really cool. That's my goal.
Do
10
you
years,
guys, mm,
go ahead. I'm sorry.
do you guys know a meeting called ARDD in Copenhagen?
Mm.
I read something a little about it, but I didn't
Yeah.
know too much.
If you search this ARDD, it becomes the world's largest conference on aging, drug development, and as well as companies, the startup companies on aging research, on commercialization of anti-aging drugs. It, um, it has its annual meeting majorly in Copenhagen. So I strongly recommend you guys to go there next year because it's such a wonderful opportunity for you to expose yourself, make networks, talking to other people who are on the same path of you. You can learn something from them. It's just a wonderful one. Because both of you are interested in commercialization, then I want to share with you one example I got. I think I'm very lucky, not only just to do basic research, but I've been very actively to translate like benchtop research to bedside. For example, in Norway, it's a country very famous for its sea food. There's also a small, actually big place in Antarctic belongs to Norway, whereby we have a type of fish called krill. It's one type of tiny shrimps. And we collect a lot of these krill and ice. And it's not a way, it's like there's a company in Norway has a patented way to extract the nutrition, especially the oil, from the krill. So the Norwegian-based krill oil is there. They have a spoon, have it daily because we are shortage of sun exposure in the winter times and a lot of tradition. And I have been working with this company for a few years. They published a paper showing this krill oil can reduce the wrinkles in the roundworm C. elegans, makes them living healthier and longer. Then a company, which is a joint company between China and Norway, they recruited me as a chief scientific and medical officer. And now this company with a sales revenue of $80 million per year.
Wow.
Yes. And I also like it's a new product we released recently. I think it's beautiful.
Mhm.
The background, do you know one guy? He's one famous Norwegian artist. His name is Monk. So who has if you search Monk, who was a very world famous artist, and the background is from the Monk's representative painting called Screaming or something. Yeah. So if you are interested to build a company, so it's not one people's thing, it's a whole team. I think for me, I'm very interested and energetic for this company. First, I need to make sure the product really safe. It is from Norway, there's quality control. And secondly, it really works, at least in animals. In humans, we do not have the same, but at least it's safe and works. So it sells as a diet supplement, not as a drug. And number three, which is you need to have a team, how to marketing. If you look over this one, I think it's beautifully designed. It just make you happy when you have a small bottle like this. So it's all the things together to. Since I talked to a lot of young people like you guys who wanted to open a company, actually it's a very nice idea, but the road is challenging. Not so many people can be successful, especially to earn the money. That take actually a couple of long time. How can you convince people to invest on you?
Yeah, being an entrepreneur is all about selling yourself.
Yeah,
It's like 20% the idea, it can be awesome, but if the team and who's running the team, like say I'm running the team and Heem's on my team, but we don't really get along, no one's going to want to give us money because the idea could be great, but if the team can't run it, there's no point. They just waste their money. So you have to really build a good cohesive team around a good idea to really have a chance of success. And that's something that I've learned, day in, day out being up here at school and not only talking to scientists who have commercialized products, but also just general entrepreneurs who have come in here and created, whether it's pieces of tech or it's like a marketing firm or just something. It's all about the team. It's all about the people you surround yourself with. And, when we talk about your lab, it sounds like you're just surrounding yourself with great people, both, just at your benches and all around the world with, people from Denmark and people from England and your connections back in the United States and even, back probably in Asia and
also in China. Yeah.
yeah. And it's, it's incredible. It's really incredible to see even what you might feels like a small network of people. It could be 15, 20 people, but like you could like legitimately change the world if you guys find something with aging and a cool small molecule that works, and all of a sudden, you're off and running and you have a great company that's going to help a lot of people. And I think that is just like the coolest part about being in this program and being in this field is, the sky's the limit. And we all really are working for like a good purpose. So even from the krill oil marketing to anti-aging through Mitophagy, like it's all so great.
Yeah, I just want to add one more thing. When you run a company, so everybody wants to sell their products, in the best way, but you need to be absolutely make sure your product really works. You should also have a balance not to overselling your thing. If you use Elizabeth Holmes, right, the lady from Stanford University, blood can test give you everything. That was a major joke in the field because she oversold sells the same. Then she become in a bubble of herself, using one excuse to cover another one. And I think for me, especially a scientist who wants to have my reputation stay here for long, I need to absolutely make sure the paper published in my group can be replicable. The things I said, so I have a very strict contract or agreement with the company. You cannot say the things which it does not has been tested. You only can say this is in C. elegans, it can reduce the wrinkles. You can't say it reduces wrinkles in humans. We just need to be, especially in a country like Norway, we are so transparent, isn't like a trust-based society. If you lose your trust, then it's hard for you to survive.
How did the COVID vaccine go down in Norway? Just, I know this is kind of out of left field, but we talk about compliance with, the people from Norway, and you guys are worried about, the quality of the products that you guys are putting in your bodies and the food you're eating. I know, and Heem and I know from personal experience in the United States with the FDA, some people would argue that the COVID vaccine was rushed, and some people were hesitant to take it because of that. How did the Norwegian, how did the Norwegian public react to COVID and the COVID vaccine?
So definitely, I think the good thing in Norway is the politicians do not give too much influence. It is scientists and the society, they tell you whether this vaccine, for example, COVID, you must have a vaccine to stop it. So this is, I think, the mainstream media as well as TV in the country is they listen to the scientists and they listen the recommendations. I can't give a lot of comments, but from my experience, very simple. The university purchase they believe which are qualified vaccine. Then they ask us to do the injection. Then we accept. It's very simple, not too much conflicts or doubts.
Thought it should have been over here. Ridiculous. It was kind of ridiculous, but I was just curious because people from the United States would feel like we're in like our own little bubble, and the rest of the world doesn't exist sometimes. And I've never heard anybody else's experience with like the COVID vaccine in 2020, 2021.
Yeah.
But I just thought that was interesting and kind of on topic because it's gene therapy and mRNA. So that's all. Thank you.
I think it's also important to like note how a scientist explains a concept is so important in public trust, because if they're saying a lot of scientific terms that people don't understand, then it's kind of like you're using your knowledge to like overpower somebody. But the way you, like, for example, the way you explain mitophagy and how the garbage truck is losing its engine's power, and we have to try find something that gives the engine back its power so we can continue to take away the garbage, and then how this helps your long-term like health and everything. That's so simple, then that can be understood by anybody, that like I feel like giving you any funding is like the easiest thing, because you made it so easy for anybody to understand.
Yeah.
So I feel like that's a huge props to scientists like you that make that so easy to understand for everybody. And yeah, so thank you for that. And like we really like wish the best that all like your research does like amazing things, and we're really interested in like keeping up with your journey for sure.
Definitely. Just so on the 18th to 19th, I'm going to co-host the first Norway UK meeting on aging and dementia with my colleague from Oxford and King's College London. Actually, the program will be streamed through Zoom. I will also send you the link just in case you are interested. We have the top leading aging researchers to give the talks.
Absolutely.
Okay, good luck with
Very,
that. That sounds
very
awesome.
interesting. Great.
Good.
Send that over our way. But all right, well, we want to thank you for coming on, Dr. Fang. It's been unreal to talk to you. given a lot of insight on aging and dementia. And we just want to say congratulations on all your work, and we wish you the best of luck in the coming months and years. But to everyone watching, we want to say thank you for watching. We hope you enjoyed. Look up Dr. Fang on Instagram, look him up on Twitter, everything. Just unbelievable stuff. So yeah, this has been another episode of Cells to Cell. I'm Nick, and that's Heem, and we'll see you next time.
Thank you so much, Heem and Nick. Yeah, see you next time. Bye-bye.
Thank you.
Yeah.